I'm Claudia Nischwitz. I'm the extension plant pathologist and also run the Utah Plant Pest Diagnostic lab. And I'll be talking briefly about a few new diseases and insect pests that we found over the last couple of years. So Fusarium wilt on ranunculus was first time a year and a half ago that we saw that. The host is ranunculus, and this fungus is very host specific. This strain will only go to ranunculus. You might see Fusarium oxysporum infecting other plants like vegetables, but these will always be very specific strains. So the one that goes to tomatoes will not go to ranunculus and vice versa. The symptoms you see on the plants is that the plants start to wilt. Initially, when it gets cooler, they might recover, but over time, they just permanently wilt. If you cut the stems and the corms, you see a vascular discoloration. So you see the vascular bundles in the middle here, they turn brown. They should be nice and white. So that is an indication of an infection with Fusarium. The fungus is introduced in infected corms. So it's always good to buy certified disease-free corms if they are available. There may only be a couple of corms that are initially infected, but over time, the fungus will move from plant to plant, especially if you plow and till old corms under and they get chopped up. The fungus can overwinter or survive for a while on the decomposing corms, and then it just moves through the roots and going to find new roots and start infecting the new plants. Management, you want to completely remove and destroy infected plants, including corms and roots. You do not just want to plow them under and then plant again ranunculus on top of it. You need to rotate with non-host crops for several years, at least 5 or 6 years, to reduce the amount of inoculum that's present in the soil. And to my knowledge, there are no resistant ranunculus varieties available. Coneflower rosette mite, that was an interesting one we saw last year from Melanie's garden. So coneflower rosette mite is an area-feared mite. It's so small, it's a lot smaller than a spider mite, so you cannot see it with the naked eye. You need a microscope to see it. They spread to new locations by the wind, so what they do during warm temperatures and strong wind, they crawl up to the tip of the flower or the leaf and let the wind take them away. The mites then feed at the base of flowers they colonize, and the symptoms you see are rosette-like tufts on cones. And sometimes they are green, so they can be mistaken for infections with phytoplasma, which are bacteria that do not have a cell wall. So here we have some pictures, and you can see the cone flowers, and you see those green, almost little leaflets that are coming out. And here that's an older one where you see them coming out as well. And so they almost look like extra little flower shoots trying to come out. So we took a few pictures with the microscope. So this is a 65-time magnification. If you look very closely where that tip of that arrow is, you see a worm-like creature. And then here on the right, that's a little bit more magnified. You can see those mites again. But you need to magnify it at least 65 times to see them. Management, cut infected flower heads as soon as symptoms are detected before the mites get a chance to migrate. And ideally, bag those infected flower heads and then move them into the trash. Do not compost them. It could give the mites a chance to get to the top of the compost pile and then let the wind take them away again. Dry bulb mites, I thought I'm going to include them. We did not see them on actually a cut flower. We found them on garlic, but they can go to tulips and other like ornamental allium species, so I thought it was worthwhile including them. They're also area-feared mites. Their name is Eiseria tulipa, and they cause red to yellow spots on the outside of tulip bulbs. Infested bulbs will dry up, and sprouting and root development of tulip bulbs are inhibited. And again, if you look very, very closely, you might be able to see the mite sitting on the garlic here. Management is difficult. Low temperature storage is the best option to reduce the mite, but it might negatively affect tulips. Early planting is also an option, but otherwise there's no chemicals you can apply to control those mites. And lastly, we had cucumber mosaic virus on coneflowers. Cucumber mosaic virus has a very wide host range. It has over 1,200 known hosts, including cucurbids, like cucumber, melon, squash, pumpkins, other vegetables, tomatoes, peppers, some cut flowers like dahlias, and a lot of weeds, nightshades, curly dock, Jimson weed, just to name a few. The symptoms you see on the plants, usually on the leaves, is that yellow mosaic and ring spot pattern. Those plants were in Alex's trial. I don't know if they had any effect on the flowering of those plants. We'll ask him when the question session is on. So cucumber mosaic virus is transmitted by aphids in a non-persistent manner, which means every time they want to transmit the virus, they have to feed on an infected plant again. They have a few virus particles on their stylet, and then as they insert that stylet into the host plant to feed, they deposit the virus particles. And so they slowly move the viruses from plant to plant. Monitor for aphids to manage cucumber mosaic virus. You could put out yellow sticky cards or just use a Tupperware container and tap plants to see if you get aphids on these plants. Sometimes they can be hard to see, especially if there's not a lot of them around. Avoid planting near cucurbids like squash or melons if that's an option. Aphids can be controlled fairly well with biological control agents like ladybugs, lacewings, and other insects. If you have cucumber mosaic virus infected plants, remove them, regardless if that's an ornamental plant in your cut flowers or if you have infected squash or cucumber plants, because the aphids will move the virus. And since there's a lot of weed hosts for this virus, good weed control can also make a big difference. And that is all that I had. Any questions?
All right. Well, thank you so much for the update this year, Claudia. There's always more that are out there to find, and we appreciate that you keep a close eye on all those.
I'm sure
Let me,
we'll find more this year,
yeah.
especially with this weird winter.
I'm curious to see how that pans out. Well, I'm encouraged to hear that with the cone flower that the heads can be bagged. I don't have to dig up my flowers and start over or anything like that. So that's good to hear. There's a couple questions coming in here in the Q&A. If you have more, please feel free to add them in. But the first one is, can your lab test trees for fire blight? The pear
Yes,
trees, oh, the answer is yes. She said the pear trees in our orchard look like they have it. I would like to confirm before selecting a treatment.
yes, we can do that, but we'll have to wait until the shoots start to sprout because we need to wait for the bacteria to become active.
Okay. Maybe could you just let everyone know that when what they need to do to send a sample to your
So
lab?
if you want to send a sample to the Utah Plant Pest Diagnostic Lab, we need new shoots that are starting to show symptoms of fire blight. Usually when the blooms come out, a couple of weeks later, you see the blooms turning brown, the leaves start to turn brown, and the new shoot tips will wilt. Cut about a foot segment off and send that in to us, including blooms and leaves and everything, and then we'll try and isolate from those. That's the best way to do it. Older cankers, like wounds on old branches, sometimes will work, but that's sort of a hit and miss. You can also send us some pictures. Sometimes the symptoms are so distinct that you could just send us pictures and we might be able to tell you what it is. I can put our email address in the chat here.
Oh, that'd be great. I think you're the fastest at responding to emails, and I'm always impressed at how much you can diagnose from a picture itself. All right. Well, thank you for sharing about how to send samples in for diagnosis. The next question and the last question so far is, can you spot differences between CMV and TMV?
Not really. They both cause a mosaic, and it depends on the host plant because the symptoms on individual host plants are not always the same. So ideally, you send a sample to us and we can do some testing to determine which virus it is.
Thank you for that. And so we've wrapped up on questions now. Alex is on, I see. I think you mentioned him during your
Yeah, I was
talk.
curious if he saw any negative effect from the CMV infection on his coneflower production.
So we didn't let our flowers go into bloom for our establishment year, so we did not see any differences in the cones themselves. But going into this upcoming field season, that's where we may be able to observe if there's any differences with the virus itself.
I think that would be good because some viruses do affect blooming and they can cause sometimes distortion of the flowers. You might get color breaking. But since we don't know what CMV does on a coneflower, we'll have to wait and see. It may not have any effect at all, and it may just be, a fluke on the leaves.
Well, I'd be happy to share anything that I find for this upcoming field season. So,
Sounds good.
We set out to test one or two things, but then each study we end up learning so much more because of getting to work with each other and so forth. So so everyone stay tuned. We'll report next year on what happened. Are there any, so here's another question for you, Claudia. Are there other ranunculus diseases to watch out for?
Ranunculus can get tomato spotted wilt virus. That's a thrips transmitted virus. And the thrips can transmit the virus for their entire life. So they have to acquire it as larvae. So they have to grow up on an infected plant. And then as adults, they move it from plant to plant. And they can live for about 3 months. So they can spread the virus for a long time. For thrips, there are some insecticides available that you might be able to use on cut flowers, but you would have to spray them early on. Thrips reproduce mostly parthenogenetically, which means you have a female but no males. And one female can lay 200 eggs, and they take about 3 weeks from egg to maturity. And then each of those 200 new females can lay 200 eggs. So you can do the math how fast you can get an explosion in thrips. So you want to catch them early, and sticky cards are really good. Yellow sticky cards or blue sticky cards. And then you just need a hand lens and you can see these orange specks of dust on the sticky cards.
So pest and disease management really do go hand in hand.
Yeah.
Awesome. Are there any other questions here for Claudia or anyone else? As you're adding the last questions in, we do have an announcement. When Molly Payne was giving a talk on behalf of the UCFFA, she brought up how there was a bill being discussed this morning about cut flower farming. And so we wanted to update you that that was passed unanimously by the committee. And so if anyone's interested in following this case, we have posted a link in the chat for you to do so.